All right, hi everyone. So it's my pleasure to be here to talk about Vera Therapeutics and also this new FDA approval for IgA nephropathy new drug. So we have already talked about TACI-Fc. I think Charlene is here, and then we have been talking about this for a couple of times. For example, Alpine. So, why this news is very exciting because this is the first FDA approval for TACI-Fc and also the first approval for this target as a first targeted TACI therapies. And TACI has been discovered for over 20 years and, from contribution from both academia and industry. And also Charlene mentioned Genentech has also contributed to this field. So today I just want to briefly summarize for this field and why this project is interesting and what it might bring to us for the future for B-cell targeted autoimmune disease and beyond. So TACI actually is a receptor. So together with the ligand BAFF-APRIL and also BAFF have another sister or brother, another two receptors. So three receptor, two ligand, five molecule form a B-cell druggable universe. So for example, anti-BAFF was first approved for lupus. That was 2011. And then later time was approved for lupus nephritis and also for pediatric lupus nephritis already have three indications for belimumab. So in general, anti-BAFF blockade for lupus is not very successful, although it's proved have some efficacy, but everybody knows this is not a great drug. So that's why so many people has been exploring other directions for lupus and also for utilizing this pathway to treat autoimmune disease. So beyond lupus, there are some application to wipe out B-cell tumors. For example, BCMA CAR T, we have been talking about that for a long time. And 2021, and Legend Bio and also other company, two drug has been approved for multiple myeloma. And the T-cell engager, of course, to target BCMA for multiple myeloma. And there are several programs has been approved. So all these are B-cell targeted depletion programs, including B-cell lymphoma, B-cell tumors, cancers. And there is another antibody approved last year for like IgA nephropathy, anti-APRIL. This is also the first anti-APRIL approved for autoimmune disease. So, and there's one program ongoing, anti-BAFF-R, for autoimmune disease. The idea is to block BAFF and also deplete BAFF receptor positive T cell. This is also depletion programs. So because of depletion, deep depletion by CAR Ts or even T-cell engagers, it might bring some potential efficacy for autoimmune disease. That's why last couple of years, CAR T, especially T-cell engager for autoimmune disease, has been very hot. So, for example, Candy Therapeutics will talk about that. Several companies have sold their programs to several MNCs from China, and that has been very exciting. As Leon has been talking about that for in several scenarios. So in general, how to target B cells? We have depletion approach. We also have a blockade approach. In general, depletion is much more effective, but blockade is less effective. But however, blockade, because we did not deplete all the B cells, so safety should be great. But the problem here is efficacy. Can blockade have any efficacy or surprising efficacy in one particular disease? For example, lupus, definitely, that's something people have been talking about for depletion, but can blockade have any efficacy in one particular autoimmune disease? And because of safety, right? If you did a good safety, you have good efficacy, you don't need a depletion, have a better efficacy and safety. That might be something quite interesting. You don't need to wipe out all the autoimmune disease, but some particular autoimmune disease. So that is why people has been talking about because BAFF-APRIL has been just blocking one pathway. How about TACI-Fc to block both ligand? So this is something people has been thinking about because block both ligand, whether this can do anything for autoimmune disease. One slide. Thank you, Ethan. So actually, the first TACI-Fc has been proved. Not FDA, but generally has been proved is from China, is from RemeGen, 荣昌生物, telitacicept. Early time has been proved for 2021, has been proved for lupus. Also recently has been proved last year approved for GMG, and also IgA nephropathy actually has been approved last month, and also surrogates disease. So you might be surprising why lupus, this drug from Rongchang has been approved in China, but in the US, this trial has been failed. I'm going to elaborate, but in general, Rongchang, RemeGen was a company who had the first telitacicept, the first TACI-Fc been approved, but only in China. So they have active trial in the US. So that's why Atacicept from Vera Therapeutics is the first program approved in the US FDA. So, but historically, there are quite a lot of setbacks. For example, MS, multiple sclerosis, 2009, when I'm going to talk about that story. So when I get into this field, the first MS trial was failed. Actually, TACI-Fc enhanced the disease. It might be related to BRAG. And also lupus, nephritis, and lupus also a later time has been failed. So the question here is like, what is the difference? I'm going to talk about that later. So, a couple weeks ago, July 7th, 2026, Vera Therapeutics received FDA accelerated approval for adult patients with primary IgA nephropathy. That is something we're going to talk about. One slide further. Thank you. So for this pathway, as I mentioned, so you have BAFF receptor, BCMA, TACI, and the BAFF receptor actually binds to three receptors, and APRIL binds to BCMA and TACI, and APRIL also binds to, APRIL binds to BCMA and TACI. So that's why, if you use TACI decoy, you can block both BAFF and APRIL. So then what's the functional difference of three receptors? BAFF-R has been very important for mature B cell survival, for B cell survival and maturation. So BCMA has been very important for plasma B cells. So that's why BCMA has been targeted for multiple myeloma, which is more plasma B cell related cancer. But TACI have some unique function. TACI actually important for T cell independent antibody response and also class switch, for example, IgA. So there are quite a lot of data talking about APRIL or TACI primarily regulate IgA. And APRIL have another function to regulate plasma B cell. So because of setback of TACI-Fc in lupus in the United States, so that's why some company has been thinking about some special biology. You don't have to talk about very, very severe disease. How about we target special biology of TACI to layer down the biology, to find the best application for this drug. So that's why some of the disease has been, lining up, for example, IgA nephropathy. That is why they start this program later time after the failure of MS and lupus. One slide. So Atacicept phase 3 trial in IgA kidney disease. So they have about 200 patients, mean age 40 years, almost equal sex, male and female. So this drug is actually they use a sub-Q 100 mg dose, but be careful. This is once weekly self-administered auto-injector. So it's a sub-Q injection, has to be injected weekly. So because there are some kind of proteinuria, surrogate data compared to placebo, which is literally have low effect, have more like 45% reduction after the drug. So that is giving some confidence by the FDA. So that's why it get approved. And another thing is safety profile is very interesting. So any adverse effect compared to placebo is more like 59 to 50. There's no very severe grade 3 and 4, so that's why this is a very safe drug. Even we talk about some of the immunosuppressants might have some infections, it's really very similar compared to placebo. In general, this is a very safe drug. But however, for this trial, they have some problem like kidney functional data. For example, EGFR or something, which really reflecting the kidney filtration function has not been reported. It will going to be coming out next year. But in general, because surrogate data already good enough, that's why FDA very quickly approved this drug. One slide. So compared to anti-APRIL or approved last year, I would say the proteinuria data looks very similar, 51 or 45, although there is numeric changes, but in general, because this is a big trial and also it's not head-to-head, in general, people will consider as similar. But however, anti-APRIL already show some data for the kidney function. So of course, we have been waiting for the atacicept data for the kidney function. In general, anti-APRIL is no worse than atacicept or at least similar. But the real data is coming. We're going to be determining that whether kidney function, which one is better. But from the data, and also I was going to mention anti-APRIL do have some advantage. For example, this antibody half-life is much longer, but anti-APRIL also have some limitation. Of course, only block APRIL, but the later kidney data we're going to be very critical to determine which drug is better. One slide. So then, besides Atacicept, there is actually several programs. I mentioned about telitacicept and also Alpine. We talk about several times. Povetacicept. So there are three leading programs already entered phase 3. So TACI actually is a very interesting protein. Has two CRD, 1 and 2, and actually TACI have N-terminal peptide. So N-terminal peptide is not involved in the BAFF-APRIL binding. So that's why people has been focused on CRD1 or CRD2. That's why atacicept is basically CRD1 plus CRD2 make a fusion. So this is a picture in the middle. CRD1 and CRD2 make a fusion to block ligand BAFF and APRIL. So nobody knows what's the function of N-terminal peptides of the TACI. That's why, and another thing is CRD1 and CRD2 are very redundant. They actually combine to BAFF APRIL, but they cannot engage with the ligand at the same time. They either bind to CRD1 or CRD2. That's why povetacicept, they choose to, just incorporate CRD2. Instead, they make a mutation to make a much more better affinity. So that's why, as you can see, the three programs, although they are both TACI programs, but they are different. So telitacicept have N-terminal peptides. Nobody knows what's the function here, and nobody knows why Rongchang Sengwu want to incorporate this N-terminal peptide. And tacicept is the very first version, which incorporate the only ligand binding domain. As I said, povetacicept from Alpine is a shorter version, but have mutated. So then, which one is better? One slide. So then also the question here is like why Rongchang Sengwu RemeGen want to incorporate that N-terminal peptide. So in general, the binding of the povetacicept, this is from Alpine. And a couple years ago, 2 years ago, we talk about Vertex acquired Alpine Biosciences with $2.9 billion, mostly for this program. So povetacicept is a shorter version, only incorporates CRD2. However, it can block both APRIL and BAFF in a better way compared to a Y-type TACI, which is a tacicept, or even the Rongchang Sengwu's RemeGen's a longer version. So the whole field consider povetacicept from Vertex or Alpine is a better version, is a best of class in terms of blocks the ligand. And as you can see, especially for the APRIL, the affinity is much higher to bind to APRIL and also block APRIL and BAFF mediated interaction with the receptors. So that is also the reason why Vertex spends so much money to purchase this program. One slide. So then, what's a comparison of those three different programs and also anti-APRIL? So as I said, this is anti-APRIL is antibody. So CMC is better, and also the half-life is better. So it's a 400 mg subcutaneous every 4 weeks. Atacicept has been approved, but it's more weekly injection. And telitacicept from Rongchang Sengwu is also very similar, weekly injection. But the povetacicept is shorter, and also they can do every 4 weeks. So half-life is better. This is CMC is better. I cannot say CMC part, but I think the half-life part, PK part is better. So from the data, especially for proteinuria data, looks similar, but the pathogenic biomarker data, I would say, um, look numerically speaking, I think povetacicept looks better. But the telitacicept from RemeGen, I didn't find the final data, but I think from the some of the other sources, I found it's roughly 50%, maybe a little bit lower. Maybe again, all of this needs better data from the kidney function. That's number one. And also anti-APRIL do have some advantage in terms of half-life, and also povetacicept. Again, everything will going to be settled after the kidney data. One slide. So then the key question for the TACI-FC field, I think this is very exciting because anti-APRIL kind of approved data in IgA kidney disease looks good, and also this is the blocking data. And also TACI-FC now has been proved is very safe. So this proves that you don't need to deplete every T cells. You just need to block some of the key pathways to reach the efficacy and the safeties. So and as I said, safety profile are better and are over B cell depletion programs. So for some specific disease, you do not have to deplete these B cell. That is the take-home message. You can reach very good efficacies. This I would say anti-APRIL and TACI-FC will make this blocking agent very interesting to some of the autoimmune disease. So we do not need the B cell depletion for some autoimmune disease. That is a take-home message. But another thing is TACI-FC program better in kidney function in IgA kidney disease versus anti-APRIL? I already mentioned that. That is a very important message in later time. So how about other indications beyond IgAs? Because MS field, lupus has been filled. How about other indications? There are several indications already ongoing. For example, lupus nephritis and surrogate disease, for example. But why atacicept has been filled in MS and others? This is everything is talking about IgA or talking about B cell, but why approve some autoimmune disease, but and has been successful on others? So we really need to understand the biology, and also because atacicept has been failed in multiple disease in lupus, but why telitacicept from RemeGen has some efficacy in lupus in China, and whether this data can be expanded in US, but why atacicept has been failed in US? What's the difference? Is that because of the drug difference or because of patient difference? This is also open question because RemeGen already have several indications approved in China. This is a very important question, especially RemeGen has N-terminal. This telitacicept has N-terminal domain of TACI. Does this N-terminal domain of TACI has any functional meaning? That is a very important question. Nobody knows. So then I have two slides, one slide more. So we just have a paper published. This paper coming from my postdoc studies. I started this project 2011. Right now it's already 15 years. I really spent lots of time, and unfortunately we made this to today get it published at Immunology. So basically we found the third ligand for TACI as a first inhibitor ligand. As I said, BAFF-APRIL is a ligand to activate B cells. It's a first inhibitor ligand for B cell through TACI. FGL1 is so far the only ligand, only binds to N-terminal peptide of TACI. And the function is not to block the BAFF-APRIL, is to make TACI endocytosis to reduce TACI levels. So that's why FGL1 have some function to reduce autoimmune disease. And very interestingly, our paper was accepted July 8th, one day after FDA approval of Vera Therapeutics Atacicept. One slide. So then the basic data is there is also very interesting that only human TACI have this 1 to 30 from MSG to VAM from this 1 to 30 N-terminal peptides. Mouse do have some, but mouse binding is through a little bit different locations. but human have very unique N-terminal peptides. If you check other species also have these peptides. So nobody knows what's the function before this paper. So we found FGF binds these locations through charge interactions very different from BAFF-APRIL. So that makes FGF as a third ligand for TACI and the only ligand which engages with N-terminal peptide of TACI. And it really took me 15 years to finish. And also FGF1 is only ligand for TACI. And before we talk about FGF1 also binds to LAG3 from my another paper. And very interesting that TACI now approved. LAG3 has been approved. So, both receptor has approved targeted immunotherapies. So then to conclude, I would say TACI is getting very interesting. It has been very cold when I started to work on that, 15 years ago. But now I think it is on the stage. I hope there are more future TACI-based better therapies for autoimmune disease or other human indications.